what I'm going to do today is really to bring you back to the very beginning of the research process. Because in this meeting, and we started already with Dr. Betlak, we will talk a lot about clinical trials that have different phases, and we will learn about the phases and the process of the clinical trials, whether we test for safety or for efficacy, and how to design better and improve clinical trials, including outcome measures, as Rick just told you. And clinical trials that really represent the most known component of the research progress that you are all already very familiar with. But in reality, these clinical trials represent really the very last stop of a very long process that involves clinical research in general, which is not linear, as you will see in the minute. And Alison really asked us earlier, she told us why she's here and why we are here. I guess that the answer for me is really that as a researcher, as a bench researcher, as a lab researcher, I'm really here because I'm very passionate about educating the patients about this process and educating the patients of why we don't have yet an effective treatment, and telling them really try to again talk to the patients about the challenges that the disease itself brings to all of us. I always give an example. I am a double melanoma patient, and even though I am a researcher, when I go to the clinic and see the oncologist, I always ask questions, and I never want to why there is not yet an effective treatment, why this didn't work. And I don't know the answer, even though I'm a researcher. And so I think it's always very important that the physician or the researchers get back to me with very basic answers, and they're always honest. And oftentimes, as in the research field, in the basic research field, we are really lab mice, right? We are a little bit more introverts, and we tend not to really go out and share our experience and involve the patients and the community early on in what we do, recognizing, however, that it is incredibly important. Research, whatever it is, starts with the patients and then with the patients. And so in our center, for instance, I work very closely with Dr. Ilieva, and the research, the lab research is always present in clinic to answer any question that patients may have. So I really want to thank all of you for listening today, and for also your eagerness to learn about the first step of the process. Every treatment on the market is really the results of years of research, not just studies involving the treatment itself, which are the clinical trials, as we've just said, that also requires a lot of time, but also the research into the disease or the research into potential drug targets, and also drug development, which normally are tested in preclinical trials. This is all described as clinical research, or better yet, as translational research, which is really the research process that applies the knowledge from the basic biologies, from the bench, and the clinical trials to techniques and tools that really address critical medical needs. Unlike applied science, pure basic sciences, translational research is specifically designed to improve health outcomes, and really aims at moving findings from the bench to the bedside, translating this basic research into ways to helping patients. And so today, because I'm really passionate, as I said, about educating about this challenging and difficult process, but necessary process, I will very briefly and in general terms walk you through this first step, the basic and translational research. I will tell you why we think basic research is important. What are we looking for when we perform this first step in the translational research process? What are the tools that we use? How do we use these tools, and how do we refine these tools as we acquire more and more information? What are the challenges that we are facing? And because, as I said earlier, this is not a linear process like I showed you in the previous slide, how we do redesign and refine our research. We learn every time we end the clinical trial, we learn something, and therefore the clinical trialists, my colleagues here on this call today, come back to us and tell us, listen, we have learned this and this and this, perhaps it's better that you redesign your research and you go and look for X instead of Y, because this is where we think that what we have learned from the clinical trials. Vice versa, it is imperative that every time we have a little findings in the laboratory, we go immediately back to our clinical trials colleagues and tell them, perhaps this information is going to help you at the molecular level inside the body or inside the cells to really redefine how you choose your drug and how you design your clinical trial. This non-linear process that obviously ends with the ultimate goal, which is to find the right treatment for the patients, and I will go even a step forward, the right treatment for the right patients, and I will get to that in a minute. So why are the first steps so important, and why? I would like to stress that every time we go back to the bench, we are learning something new, and therefore it's not a disappointment to go back from to the bench, provided that we do it quickly and we do it targeted really. In fact, it's not a going back, but it's really a moving forward. Because every time we go back to the lab, it means that we found the key information about this complex disease, or a new technology allowed us to make better tools. And I will show you some examples in a minute. Most importantly, why are these first steps very importantly? Because the disease itself demands that we go back to the first steps, and demands that we, because it is a complex disease, as you will see in a minute, demands that we study it systematically and carefully. Always, as I said, revisiting our previous findings and always asking why a trial did not work, how can we do better? Should we look at the disease from a different angle, because the angle that we have been looking for so far perhaps wasn't the perfect one. Now, we all know the urgency, and I want to stress that asking this question is not an academic endeavor. We know that we work for you, and we all know the urgency of it. And that's why we would like to involve you from the beginning, very early on. It is really listening. This exercise is not an academic endeavor, as I said. It is really listening to the disease, which unfortunately so far did not respond well to all the therapies that we've tried to design. It is listening to our clinical trials colleagues, and importantly, it is listening to the patients. And so, if we start listening to one another, we also have to listen to the disease itself. The ALS, unfortunately, is an incredibly complex disease that we all know is not really created equally. And therefore, our goal is to treat the patients, the one therapy fits all approach perhaps may not work. We all know that ALS is heterogeneous, as we say, at the clinical, at the genetic, and also at the pathological level. I don't have to tell you, and I won't go into the details about it, but obviously patients come to the clinic with different site of onset. We can have limb onset patients or patients with bulbar onset of the disease. The duration of the disease is very variable. We have very fast progressive patients, very slow progressing patients, and everything in between. We very well know that ALS is part of a spectrum of diseases that also involves frontotemporal lobe dementia. And therefore, the complexity and the heterogeneity of the disease really is broader than we thought 25 years ago when I started to work on ALS. Genetically, we really know that 10% of the patients are familial, meaning that there is a gene associated with that causes in the family that causes a defective gene that causes the disease. And yet, even within this 10%, there is not one single gene that causes ALS to, again, increase and broaden this heterogeneity. But we also know that 90% of the patients are what we call sporadic. It happens once in the family. Whether we haven't found the gene for these single patients, more the sporadic patients, we don't know. But this is the complexity. To increase the complexity, we really do not know what is the beginning and where is the beginning, what causes the disease for the majority of these patients. The heterogeneity that we do see clinically and genetically is also seen pathologically in postmortem tissues from patients. We do see that patients, depending on the kind of disease that they have different pathologies. So all these differences lead us really to believe that perhaps ALS is the same diagnosis of slightly different diseases inside the body and inside the spinal cord and the brain, different diseases at the molecular level, in the genes, in the proteins, in the metabolites, and perhaps also different behaviors in different cells, because we do know that although we know that the primary target of the disease are the motor neurons, we don't know that, we know that cells that surround the motor neurons in the brain and the spinal cord actively participate in the disease progression. So we need the research really to find out who these different molecules inside the bodies are, what they do, and ultimately we need to target them specifically in a process that is called precision medicine. Nothing really of what we do every day in the laboratory is different than an intelligence approach to find an enemy at war. And if we all think that in this call we are all collectively at war with ALS, then the job of a bench researcher is really to find out where the enemy lives, where it hides, and how it functions, so we can really surprise it and kill it right there. So, and like in many wars, unfortunately, there are still challenges that, however, I really believe that with new technology and refined research every day and thinking out of the box and involving you guys with the samples that you can give us every day, we can overcome some of these challenges. And here I've listed what I believe are two major challenges. There are many others, but I would like to focus on these two in particular, the first one for today. A big challenge is the fact that contrary to the cancer field, for instance, where researchers can really study the cancer, the specific cancer itself, right? We do a biopsy or we surgically remove the tumor, and then in the lab, we can study the specific genes, the proteins, the metabolites, the composition of that tumor to target it specific with the chemotherapeutic agent, which is really tailored to that target. We cannot, in ALS or in any neurological or neurodegenerative diseases, we cannot remove the brain or the spinal cord of a living patient. And even when we do a spinal tap to collect the CSF of the patients, this is really not, as you all know, an easy and pleasant procedure that can be done many, many times over time, so we can understand how the disease progresses and what are, again, the key molecules that make you progress fast or slow or in between. So as I said, I will focus on this challenge number one today to give you an example of how research and technologies we are working on can help us overcome this challenge. But we have to remember that a major obstacle will always be for us the fact that we are really trying to bring back the motor neurons in particular that you can see here, something that may be already be dead. And so we have to be smart and catch through a systematic analysis of the basic research, be smart and catch these motor neurons before they are dead, or at least while they are still not doing well, but they start to degenerate. So we can really stop the neurodegeneration process, the degeneration process of the motor neurons right there. So they still may not function as they will normally function in healthy people, but at least they function somewhat that can help any single patient walk, yes, perhaps with a cane, but still walk and stop the gradual degeneration that will eventually make them die. So what are we looking for when we do this first step of lab or translational research? We are looking for two major categories of molecules, which we call biomarkers and therapeutic targets. Biomarkers that in medicine is really a measurable indicator of a presence or the severity of the disease. We are looking at markers that can tell us very quickly and not by exclusion whether you have ALS or not. And very importantly, we are looking at markers that can tell us and you, the patients, what kind of ALS you may have. We are also looking at markers, for instance, that can tell us if at the day of diagnosis, it can really help us telling you whether you have whether your disease will progress fast or slow. We are also looking for biomarkers. We have heard Dr. Bedlak a lot talking now about this screening in terms of trials, etc. What we call about pharmacodynamic biomarkers, right? For us, in order to understand whether when in any given trial, a drug is really getting into the brain, whether it does what it is supposed to do, we are looking for those molecules that will really tell us, let's say that they go up in expression or function, that function changes as disease progression, but with a certain drug, we are trying to knock it down or to make it work better. We are looking therefore for a marker that really tells us the drug went into the brain and in fact target that specific target that we aim at, and it brought down the expression and the function, and yet it didn't work clinically. Or maybe it didn't work clinically not because the drug was wrong, but because it didn't go as we wanted into the brain, and therefore it didn't hit the target as we were hoping. And there again, once we find this biomarker and we have this answer at any given during any trial, then we can go back and design a better way to deliver that drug and therefore target the target and perhaps having also an effect at the clinical level. We are also looking for therapeutic targets. And because, as I said earlier, ALS is a heterogeneous disease, we are looking perhaps at therapeutic target. The combination of biomarkers and target will help us identify therapeutic target specific for each subgroup of patients. Any gene can be a target. Any molecule that is activated or deactivated by this gene can be a target. Any cell, remember that I told you that although the motor neurons are primary target in the disease, we know very well that within the brain and the spinal cord, there are also other cells that are bad guys that actively kill the motor neurons. And therefore, if we identify something that can really make these bad guys turn into good guys, perhaps this is also another good therapeutic target. So how are we doing that? Lab research really works through models, whether cells in culture, yeast, or very small animals like the worms or the fruit flies, or larger animals like the mice or even the monkeys, the non-human primates. Each model has its own advantages. And the trick is really to use each model to our benefit. What do I mean by that? The trick is really to extract from each model the right information. Sort of like putting a puzzle together. Each model can be a piece of the puzzle that we aim to put together by comparing the results that we get from each model and putting them together, each information from each model, so we can make the larger picture. But of course, the best model is really the patient. And so despite the challenges that I just told you before, right, we cannot really take the brain or a piece of a brain or a spinal cord of a patient at the day of diagnosis. Despite those challenges, now we have new technology that is giving us the opportunity to study the patients, and even those patients that are not familiar, for which we don't have a gene that we can put into any of the models that I just described, and so recapitulate the disease by using the defective genes. And I would like to draw your attention to two particular technologies that we are using more and more now, that really gives us hope that although not the real brain and the real spinal cord of a patient, we can get really to some information about at least the molecular composition of each individual patient. One is really the brain-derived, the patient-derived brain cells in a dish. We use stem cell technology to really make in a dish your motor neurons, the motor neurons of the patients. How do we do that? We have now the ability to take either the skin cells through a simple skin biopsy, or even better, the blood cells from a patient by just drawing the blood in clinic, and then going back to the lab and really just with simple recipe like cooking really, convert these blood or skin cells of the patients into the stem cells of the patient. We know that the stem cells are pluripotent. They can become anything, any organ, any cell in the body. And therefore, again, with few tricks that we have in the lab, we can now take the stem cells and convert them into the patient's motor neurons, or even better now, and I will show you in a minute, even better now, organoids, mini brains of the patient. Now, is it this as good as really getting the real spinal cord or the real brain? No, but it is as good as it gets. Why? Because now I know for a fact that at least, even though these are new motor neurons, they are not adult motor neurons, I know that these motor neurons that I made in the laboratory, starting from your skin cells, contain all your genes, all your proteins, everything that makes you as a unique patient, and everything that makes you as a unique, perhaps, ALS patient, or that makes you a bulbar patient versus a limb onset patient. I spoke also about another challenge, which was the challenge for us to study disease progression by doing a lumbar puncture and taking this cerebrospinal fluid of a patient over time. Now we know that we can also overcome that challenge in the lab or in the clinic by starting with the clinic and taking the blood, for instance, of the patients. Why? Because through research, we really found out that there are these things that are called extracellular vesicles that are released by every cell in the body, including cells that are part of the brain and the spinal cord. Technology now allows us, and for each cell releases these vesicles which contain specific markers of these particular cells. So we know through technology and through research that, for instance, liver cells deliver, yes, extracellular vesicle that contains something that is specific to the liver, while the brain releases this extracellular vesicle in the periphery, in the circulation, in the blood, but that are specific, that contain specific things, molecules that are specific for the brains. Now we know how to isolate this now, and this perhaps represent, as I will show you in a second, a window into the spinal cord. So these are the possibilities now that in the lab we have that are given to us by the stem cells and by this extracellular vesicle to really study your disease, to really study the patients rather than having all these laboratory-made models. Because as I said earlier, the stem cells can become everything in our body, every tissue, every cells. And so in our case, we make them in a way that we convert them from your stem cells into your motor neuron or into your mini brain. And the extracellular vesicles which contain protein, RNA, and everything else that is really specific for the brain really represent the window into your progression of the disease. So how do we use, I'm going backwards, sorry. So I want to give you just in 2 minutes and then I'm done, 2 quick examples of what I was talking about. I told you that we can, the goal for us is to really identify biomarkers of disease or prognostic biomarkers or disease biomarkers specifically to one group of patients. Through the analysis of these EVs, now we can do it by simply taking the blood every time, for instance, you come to clinic, right? And so there are different extraction methods. I won't go through the details of this, but now we can take out of your blood every 3 months, let's say when you come to clinic, we can take your blood and then we can isolate your vesicles, the vesicles that come from your brain, right? And we can really see this vesicle. You can see here all these blue dots under the microscope. Now we have the ability, because we have markers of vesicles that come from the blood, vesicles that come from the liver, or vesicles that come from the brain. We have the ability to stain all these blue dots for markers of the brain. And once we identified among these many, many vesicles, let's say the 300 vesicles that come from the brain, we can isolate them. And then we can start the analysis. What is it that over time, as disease progresses, what is it that this vesicle for these patients or this group of patients have that increases over time as the disease progresses, or that decreases over time as the disease progresses? And we can look at the proteins that change over disease progression, or you can look at the RNA that changes over disease progression. Or we can even look at just in one time point, at what is different in the vesicles of the patients when we compare these vesicles from patients with other neurological diseases or with healthy people. And then at the protein level, and here I wanted to give you an example, we can start really to identify a group of people which are here color-coded, like in red, we have the ALS patients. In green, we have the healthy control. And in yellow, we even have a patient who is at risk of developing ALS. And based on the presence or the absence of these markers, now we can really tell you what is the ALS patient or what is a non-ALS patient. Or perhaps if this patient who is at risk will develop the disease or not. We can also look inside this vesicle, not for the protein, but for the RNA, for instance, and really start to, in an unbiased way, to divide and to tease out in an unbiased and blind way. As an experimentator, I don't know if the vesicles that I'm analyzing now are coming from a patient or from control, because I want the power of my analysis to tell me if, in fact, if I'm able to divide patients versus, to distinguish patients versus controls, or to distinguish patients in short or long-term survival patients. I can even do something a little bit further. I can just now take all ALS patients, and based on an analysis of these, their gene expression within this vesicle, I can start to stratify patients in subgroup of patients. Do I have the ability now to really predict or to say that I have a molecular signature that is specific for a bulbar patient versus a molecular signature that is specific for a limb onset patient? This is the advantage now of the extracellular vesicle. There is still a lot of work to do there, but here is just an example of how technology now allows us to really take samples for the patient, which is in an easier way for the patient first and firstmost, but also for us to really trick the system and get into the brain from really the periphery. The stem cells, and in particular the motor neurons or the mini brain derived from the patient's skin cells and from the stem cells, now allow to really do the validation of these biomarkers, right, in the real tissue, in the real motor neurons of the patients, and ultimately allow us now to study in the laboratory how these motor neurons behave or misbehave over time, and compare with your disease progression. Now, if I can look under the microscope how your motor neuron degenerate and how the molecule that make your motor neurons change as your motor neuron degenerate over time, and then compare with the rate of your disease progression, perhaps I can really identify specific therapeutic target, which is our ultimate goal, right? We want to go back to our clinical trialists and tell them, this is what you have to target, and you have to design for this particular group of patients, this trial, targeting this in this very way. Perhaps we can really now, through these cells, to really identify these specific therapeutic targets for this group of patients. And at any rate, we can really use these mini brains from the patients or motor neurons from the patient to really validate the biomarkers that we identify. Not only, you have heard about Dr. Bedlak talking about drug screening, right? And high throughput drug screening. Ideally, I totally agree. We should, and Dr. Bedlak is starting that, we should really do an high throughput screening of patients, living patients in trials. But in the meantime, we can use now these patient cells to put them in 384, 1500 in these mini wells, and really do a high throughput to screening of any drugs that targets either a specific target or a pathway that we think is shared by every ALS patient. And again, because this is going to be fast and this is going to be done in real patient-derived material, we also have the opportunity to then in vivo in the model to really test whether this drug works very quickly and how it interacts, it gets metabolized into the in vivo before we take it to the patient. But I would like to stress the importance of now having these tools that are really the tools that are really patient, actually, to really do this high throughput screening in a more efficient way. The ultimate goal, as I said, for all of us, is really to get back to our clinical trialist colleagues, to the pharma companies that develop the drug, fast and efficiently, to really tell them, what they have to develop and how they have to deliver, and how hopefully they can design trials listening really to rigorous science that has really identified something that can be meaningful. And the goal is really also learning from the past clinical trials and what even the lessons from the genetic forms, right? The antisense trials that the results have just been released, perhaps to really to recognize and to redesign really the trials, recognizing that perhaps one drug doesn't fit all, and listen also in this exercise to the results of the trials. Maybe a lot of trials that we think that didn't work and that we dismissed, in fact, they worked. But in the larger number, they didn't work for all patients, and then overall, the effect was missed. But perhaps that single drug that we use for all flavor, every color of these patients, had an effect in a subgroup of patients, but didn't have an effect in another group of patients, but had an adverse effect in another. Now, combining the results of these trials, perhaps with the molecular analysis of each individual subgroups that I just showed you in the lab, will allow us also to identify patients who respond to a trial and patients who do not respond to a trial, and therefore design the trials by enrolling patients according to this information. Obviously, there is always the personalized precision medicine approach, one drug for each group of patients. And perhaps ultimately, through the systematic analysis of biomarkers, pathogenic pathways, as we call them, and therapeutic target, even the possibility to design a multi-drug therapy with one drug individual for each group of patients, another drug individual specific for each group of patients, and perhaps to target the already degenerating motor neuron, a drug that works for every single patient. Perhaps if we really bombard this disease at every different level, from the beginning to the middle to the end, we will be able really to design a very effective therapy. And this is really our ultimate goal. And with that, I really would like to thank all of you for listening, for participating in this, for wanting to learn and become ambassador, and in particular, thank also all our patients that work with us every day to really try to get to the answer as fast as possible. Thank you very much.
Thank you so much, Piera. I know we're running a little late, but I want to make sure that everybody has an opportunity to ask a question. And Kevin has asked a question, Piera, that I think all of the faculty can comment on. You know, thank you for the education, but educating patients, you know, pals and loved ones about the process isn't good enough. What are we doing to accelerate this? And you know, I can kick it off. You know, we're trying very hard to develop biomarkers that will enable all of these targets and understanding different disease processes in different folks. To do that, we need your help. And in fact, what do I mean by that? We need samples. And what I've found is, of late, there's a reluctance to give spinal fluid. I don't blame you, who wants a spinal tap? But that is critical material. Skin biopsies to develop iPS cell lines, and you know, tissues to develop some of these other tools that can then accelerate. I think that one of the things that's been done is the development or the use really of a platform trial, which means that we can be trying multiple drugs at once and can stop for futility and put another drug in. So we're looking at 4 drugs at once. But because this is such a heterogeneous disease, we're not studying one disease, we're studying God knows how many diseases. And that's one of the complications. And I'm going to just stop there and let other folks jump in with their comments about what can we do to accelerate the process. We understand the urgency, believe me. Nobody knows more than me how urgent this is.
I want to echo what Terry just said, and that's really the purpose of the education and really sharing with you the challenges, because as I said, this is really not an academic, we all know the urgency, and the urgency, your urgency is our urgency, and what we need is really these samples. Obviously, the challenges are still there, because this is what is the nature, not only of the disease because it is heterogeneous, because as Terry said, we are looking at many different diseases, but also because technology up until few years ago wasn't there, right? Now, I hope I showed you that, again, maybe it is not as good as taking the real brain, but it is as good as it gets. Now, through that technology, we are able to study the motor neurons of the patients and to look inside the motor neurons of the patients. Now, we are able not only to go to the CSF, but to go to the blood and perhaps using the blood and these vesicles as a window into the brain. So what we need now is really you to participate in this research. Many is the same of what Dr. Bedlak explained in the beginning when in 1995 he started, right? And there weren't many people that participated. What were the roadblocks in participating in clinical trials?
I should comment, Kevin just wrote that really clarifying the question. I think we are missing the point of the question. What are we doing to change the mindset of regulators and the clinicians, researchers to not simply be guardians, but advocates for change? And I think that you're looking at a set of clinicians and researchers who are truly advocates for change. Dr. Bedlak has really pioneered some very novel ways to test drugs, to bring PALs into it, and to engage remotely, do drug trials, what he described. And yes, that's why we want you here and we want you out there. We're not the people that need to be converted, but there certainly are lots of folks regular.
And that will let
officials, senators, Congress.
And that will, yeah. Terry, sorry, I cannot hear you anymore. But yeah, so I will also add that we have to continue really to.
You're not here?
I don't know, I cannot hear. Do you guys hear Terry?
Terry, you were breaking up.
Yeah. Was I breaking up? Am I still breaking up, Pat?
Yeah.
Yeah. So I'm going to shut up then and let everybody talk. Go ahead, Piera. And then Rick, you might want to say something, and Lyle.
So I agree with Terry. We have to continue to do what we are doing all together. So here we are a group of people who continue to really push for research at the regulatory level and these kind of things. And I will add that the NIH, for instance, and Terry and I are part of this group now, recognizing that funding for research and also thinking about new ways really to identify this target, to bring new and better drugs into clinical trials for all the patients. It's putting together a task force of patients, caregivers, clinicians, and researchers to really work together, and we will have to give our recommendation in October, and we just started our work. So is really a fast process to really enable really to accelerate this kind of research, to accelerate getting into approval of clinical trials much faster, and again, better designs, better research, more funding, and all these kind of things. So I think that if we keep talking and if we keep showing these new data and this new technology, and if we keep advocating for the fact that having to deal with such a challenging and such complicated disease, but we keep trying it, we keep revisiting this, and we keep having new information, I think we will get there. And we have to convince them like that. And I think we are succeeding to a certain extent.
So to follow up on Kevin's question, so I understand that, you know, to enroll patients and get the funding and all that for the clinical trials itself takes a lot of time, but is it also that the FDA and having to, the oversight of those trials, is that adding a huge, you know, amount of time to that 10 years? I think the chart said it was like 10 years to get a project through a phase 3 trial. You know, I'm just thinking if it is the FDA and like it might be here at the approval process, right? Like more as we're seeing for AMX 035, they didn't, you know, immediately respond, but they have till June to respond about that. But anyway, you know, is that an area where we should be focusing in working with the FDA and asking the FDA, since this is a critical unmet need here for the ALS people with ALS, that we should ask them to streamline their process more or offer suggestions? I just kind of want to know where we can help, and if it's with the FDA or if it's, you know, where it is that's taking those 10 or 15 years. You know, I understand on the science side, there's a lot that goes into clinical trial.
There's a whole process, you know, because right now, you know, you have to develop the drugs, and a lot of it is that bottleneck of having lots of candidates that then go into trial and having phase 1, phase 2. And remember, while it bothers us that it takes so long and that we have to go through these phases, we also have to respect safety as well as efficacy. And I think that there are steps being made, for instance, Act for ALS, and the fact that we will be having, you know, programs where you can get drugs once they get through phase 2 trials in open label studies. You know, so slowly, this kind of advocacy, what you're hearing, the advocacy for Act for ALS has made a difference. But we always have to keep in mind safety. Rick wants to say something. Go ahead, Rick.
Yeah, no, I mean, I think that these are very valid points that everyone's making. I mean, I definitely understand the frustration. I will say I've seen tremendous progress over the past 21 years in how things work in the field. There's more research now than there's ever been. I think the trial designs are more efficient than they've ever been. I think the FDA is demonstrating more flexibility than it ever has. I mean, and a lot of this has happened because of partnerships with patients. It's because previous research ambassadors took on pieces of this. Is there one place where everybody should throw their efforts against? I don't think so. I think you should decide for yourself, what are you passionate about? So Patty, if you're passionate about trying to get the FDA to be even faster, I think that's great. Try to find a group of people who share that passion, look at what's been done so far and see what you can do to improve upon it. But what we saw last year to me was extraordinary. I mean, in the beginning of 2021, the FDA put out a public statement and said they would not consider a new drug application for AMX 0035. And the rationale was, we always need 2 trials. Well, we saw a pushback from patient advocates. We saw them testify in front of the FDA in the spring of 2021, then in Congress, you know, in the summer of 2021. And lo and behold, in the fall of 2021, after those testimonies, the FDA did something I've never seen them do in my whole life. They changed their mind. They said, okay, we will accept a new drug application. Now, that doesn't mean they're going to approve it, but that's pretty extraordinary to see the FDA change their mind. Basically, that could save 3 to 4 years in the approval of that drug if they do now decide to accept the new drug application. And that's because of patient advocates. So, I mean, I think things are changing. They're not changing as fast as we would like, but that's part of why we keep doing clinical research learning institutes, because we believe that an army of empowered patients is the best way to keep improving. So find things you're passionate about. I mean, I've heard Kevin say a few times he wants more attention to patient-reported outcomes. I think that's a great goal, Kevin. Let's try to think about how we can work on that together. But if everybody takes on a piece of this, I think that's how we move the whole field forward.
I had a question or a comment. As a mother of someone who is young, diagnosed, Kate is 24 now and no longer qualifies for a clinical trial. And personally, I feel like that's a lot of data that's being lost. He could still be being studied. There are things that y'all could, or clinicians could still be learning from him. I think the restrictions on the 2 to 3 year symptom onset and many people that aren't even qualifying really hinders the advancement of science in a very efficient timeline. Just my opinion.
Andy, that's exactly what the expanded access programs are for. And that's why we're so excited to have them approved, now as Act for ALS, because those kinds of expanded access programs will enable folks like Cade to get into studies, and we will be collecting data on folks. And those are now starting to applications are being put in, and they're starting to get rolling. And, a company has to have a clean trial. We want to get drugs to market, right? In order to do that, a very rigid PALs that are tested, and it has to be, that's why these inclusion criteria are designed, because we don't want to wash out. We don't want to, as I think Dr. Pasinelli was mentioning, because we cloud the issue with a wide spectrum of with different.
You're breaking up again.
But yet we understand the urgency and to address just that issue. Does anyone else want to comment? Any of the other?
I will just back you up,
because if not,
Terry, to say that the expanded access programs are wonderful, and that the fact that we see more of them is what is giving people that are not anymore candidates for trials access. And among our patients, there's a tremendous relief that they're not cut off from trying medications. Yeah.
I also wanted to comment a little bit on Patty's question about, what are the roadblocks, right? A very big roadblock, and Rich, I think, already answered that, can be really the regulatory agencies and the FDA. But I think that we also have to be honest with ourselves, and this is my frustration as a researcher, is not that the process of developing the drug and bringing a drug to the market is like that for any disease and every disease. And the regulatory agencies do their job, right? But I also strongly believe that so far we didn't give the FDA the blockbuster drug, and not because for the lack of trying, not because we did do a difficult job, but a bad job, but because again, as I hope I convey to you, it is very challenging, and every time we learn something different. Listen, 26 years ago, when I started to work on ALS, we always said that it is one disease, that it doesn't involve any other disease. It is just a motor neuron disease. Now we know that it's part of the spectrum of diseases. Now we know that it is heterogeneous and everything else, right? So, and then with all the other challenges that we are really studying something that is in the brain and therefore it is more difficult. So my message to you is, as Rich said, yes, you have to advocate for whatever you think you want to advocate, and the help to advocate with the FDA and the regulatory agency has made a tremendous impact in and accelerated things that we do now, contrary again when I started 20 years ago. This advocacy had really allowed us now to knock at the door of pharmaceutical companies and biotechs to really, and that now they open it. When I started, there was no one who really wanted really to even invest money to develop a drug for ALS, because frankly, they were telling us there is no market, right? There are no many patients, and therefore now, thanks to all this advocacy, there are many biotechs that are working with us, even in the preclinical trials, to really develop and accelerate the development. But I think that a big component of advocacy, I want to go back to what Terry was saying, is really involvement of patients. Give us your sample. Tell patients of how important it is to participate in your clinic, not only in a clinical trial, but also in the clinical research. And tell the regulatory agency how important it is to sponsor, to fund, and the DOD, and Laya will talk about that, recognize that gap and is really putting a lot of money in that, in funding biomarker research and this kind of thing, because it is only also by advocating for that that we can really speed up the process. I think to really give the FDA something that they have to approve yesterday, because they cannot deny, right? That is really working well.
So
I would like to.
I think that, can people hear me?
You
Yes.
break up a little bit, but yeah, we can hear you right now.
We have two people who want to ask questions. I think this is really valuable. People don't mind. We will maybe not do exercise one or combine exercise one with our coffee social hour. But I want to get to everybody's. Kevin, I think you have a comment, and Ed, Mr. Beck his wife has a question. So why don't we start with Kevin? Go ahead.
Terry, you might try turning off your camera to see if it helps with your bandwidth. Maybe that will help keep you from breaking up.
So, Dr. Bedlake, I fully appreciate the idea of work on those things you're passionate about. However, as I look at how we're going to make progress, it won't come without strong support from the medical and research arm of the process. So let me give an example. From an advocacy standpoint, when Glass and Fournier wrote their op-ed in January in JAMA, calling for a higher standard out of clinical trials, who from the research or clinic communities publicly refuted that as an advocate for patients, because I've seen nothing in print nor addressed during the March MND conference.
Yeah, good point, Kevin. And again, I don't think there's ever going to be just one voice. I don't think you're ever going to get all clinicians or all scientists or all patient advocates to agree on exactly what the right thing is. But I mean, to answer your last question, I did. I wrote a rebuttal to it. It got published in, it was maybe Neurology News Today or something like that. I'm happy to send it to you if you want. If you just email me, I'll find it and send it to you. But the point that I made was that drug development in ALS is complicated. I mean, yes, there are scientific forces that, drive us toward a certain type of trial, drive us toward, replication, things that we know are hallmarks of good science. But there also has to be a humanitarian side to it. We have to think carefully about making trials more restrictive to try to find small signals. What does that actually do in our clinics? I mean, yes, if we can have expanded access alongside it, that certainly makes it more palatable. And by the same token, I mean, sure, we would love as scientists to have things replicated 5 times to be absolutely sure they're real. But I mean, there's a humanitarian cost. Each time we ask for a replication, that's 2 or 3 more years. That's 50% of all the people in the world alive with ALS that are going to die before we get the replication. So I mean, those were the points that I made. But again, I mean, we're not all going to be of the same mindset in science or clinics or even patients and families. I mean, I just think we have to find issues that are of interest and importance to us, and we have to focus on those. And if we all do that together, the field moves forward.
Thanks, Rick.
Cindy.
Cindy.
Yeah, Cindy. Yep, Cindy, do you have a question? And welcome. How are you today? You're on. You got to unmute yourself.
Unmute.
Yeah, unmute, Cindy. We're gonna have you start all over again. I'm sorry. Okay. Hey, can we unmute her?
Yeah, I had asked to unmute.
Okay.
There we
There
go.
we go. Yay. Hi, Cindy. Hi, Ed. We're gonna have you start all over again. All right.
Okay,
But we're looking
so
forward to it. Let's go.
we have
All right.
recently learned that Ed's genetic profile shows that he has a variant of uncertain significance in his SOD1 gene. And he was the first one to raise his hand to say, take my spinal fluid, take my blood, grow my motor neurons in a petri dish, and let's see if we can be of assistance in helping solve a piece of this problem. And we have been unsuccessful in getting somebody to say, I want to do that. So our frustration is we don't seem to have, as ALS patient, the access to the scientific help that the ALS world needs to provide. So how would you recommend that people in Ed's position go forward with some situation like this? Makes sense?
That's an excellent question. Thanks, Cindy. Anybody want to jump in on that?
Well, sure.
Okay.
You might start, and Cindy, with, your where folks are doing research. You might start. Can you hear me? Up. Am I still breaking up? Just shake your head yes if I am. Yeah. Okay. Maybe Lyle, you want to take
Sure.
that one?
Yeah, I mean, okay, first of all, hi, everybody who I don't know. I know a lot of you already. We work for you. I work for you. I love nothing more than these sessions because it's an honor and a privilege to be here. And what's going to happen today and tomorrow is we we want to help you be able to communicate and advocate for the specific issues that are most important to you, right? And so we all know we need patient samples, right? And we wish there was some like central way that we could get them, and someone that I could send to your house right now and say, yeah, take it today, right now, go, right? And I'll tell you, we live in this interconnected world, and we find out about things that are going on around the world immediately, right? And whether they're good things or bad things. And one of the things that I think we really need help with, and things are getting better, is learning how to harness that interconnected world for good, and really harmonizing all of these different efforts, and figuring out ways to more centrally regulate and control the flow of these samples and the flow of this data. Because you have to understand, it's not just about sampling somebody's spinal fluid, right? We need to know about you clinically. And the words that your doctor is using to describe your ALS, they have to be the same words that another doctor is using to describe someone else's ALS that's also contributing samples. Because if we're going to analyze samples and try to figure out, oh, does this one mean that you're progressing more quickly, or it starts in your arm, or whatever it might be, all that data has to be harmonized. Now, I personally, I think that there is a roadmap to accomplish that. And I think that some of the current efforts are working better than they have in the past to harmonize. And perhaps, we're going to see the CDC play a bigger role in this, I hope, in the very near future, and some of these other efforts. Stay tuned, okay? But yeah, we get it. And we know, like, if there's a way that we can help you as far as advocating and the message to get drugs into people quicker, that's great. But the other thing that I think you're hearing now, and you're going to hear more, is we need better drugs, damn it, right? Like, we have to get the right freaking drugs to the trials. We have to get drugs that are going to work, and we have to figure out which drugs work for which patients, because, like, I don't want to be an ALS expert if I can't treat my patients effectively. And I don't want to have to analyze the data 17 ways to see that somebody had a little effect. And that doesn't mean I don't want my patients to have access to things that have a small effect. But a big part of this issue is that we need to be getting the potential treatments that maybe are really, really going to make a difference, moving those ones forward quickly, and figuring out who to test them in and how to test them in. And when a trial fails, how do we know how it failed? And I'm going to talk about all this when we talk in a couple minutes. But yeah, listen, man, Kevin, we've never met. I'm looking you right in the eyes on my screen. I don't know if it looks that way on your Zoom. I'm happy to talk about this stuff anytime. Allison will tell you, like, she reached out to me about a question just yesterday. I'm like, yeah, let's have a phone call, because we work for you. And the other clinicians who are on this call, the reason I admire and strive to emulate, Dr. Bedlack and Dr. Hyman Patterson so much is that that's what I learned from them. And so your urgency is our urgency, and the goal of these couple of days isn't to necessarily argue about what's important. It's to give you the tools so that you can advocate for what's important to you. And we want to help with that. And we want to help you understand the barriers and the roadblocks so that you can do that effectively.
So if I can say something, and speaking about tools, add two things. We'll be happy to get your blood, and so we can talk offline about that. But in particular, I wanted to make all of you aware, and Jinzi, I see that she is now on the call and can talk a little bit more, I think, about that than me. But we were all at one ALS meeting in Boston last week, where we learned of a new initiative of a company, or a foundation actually, called NLOREM, right? That is called Silencing ALS. And the goal of this foundation is really to identify, the premise is each patient is unique. And unique patients do not have access to trials and to this kind of talks, because, again, N is one is not important. So identifying your gene, identifying your pathogenic pathway, and then target that through so their mission is to treat nano diseases, as they call it. So one person, one disease, and this is built and has been launched in partnership with Colombia. That's why, Jinzi, I put you on the spot. But if you want to say a little bit more, and perhaps they have a website now, this NLOREM, and I went on the website and I've seen that there is a possibility to apply, to tell your story, to say that you have this variant of SOD1, and see where it goes.
You actually summarized it beautifully. And the goal, when Dr. Pasinelli is talking about nano rare, as we look at all of ALS, and we've already talked about how there might be different types of ALS and subgroups, those groups become small. And so specific gene mutations that don't affect a lot of people living with ALS become groups of individuals that it's difficult to develop a drug for. So for those smaller, rare gene mutations, this Silence ALS program essentially partners. It goes with the idea that Dr. Astra was talking about in terms of biomarkers understanding. So one way we can identify and group people is with gene mutations. And so it's an infrastructure that's created with the help of Target ALS, and NLORM will look at people who carry the gene but don't have ALS yet. And then of course, looking at an opportunity to maybe treat those individuals with a very unusual gene mutation should they become symptomatic with ALS. And then try to develop, we're trying to leverage what we know about gene therapy platforms like the antisense oligonucleotides and try to create it and use it in those individuals. So it's another way, we can do what's called precision medicine.
Can I just add one quick thing? Just Kevin also, and again, as Rick said, everyone's got different opinions. I will say one thing that I know scares a lot of our colleagues, and scares me too, is that think about how long it takes when you go to your clinic visit, right? And think about how frustrating it is that you can't have more frequent clinic visits. And there's just not enough time in the day and not enough providers. And, insurance doesn't reimburse to improve that. And one of the things that I gotta say, I worry about, I want you guys to have access to the drugs that you want access to. But, we've seen so many treatments in ALS that don't work, right? And I worry that some of these treatments that administering something that has to go into the spinal fluid or whatever it might be, how is that going to cut into my time or my, or whatever it is to care for my own? And so I want to be able to care for my patients and treat them. But one of the problems we have right now is that if we don't figure out which treatments to give to which patients, and we're doing it to everyone, that's a huge resource suck, right? And even funding EAP, which I fully agree with and think is a wonderful thing, that takes resources to do that, and it takes funding. And I don't want to give my patients things that only work a little bit if it's sacrificing my ability to care for them and perhaps develop and accelerate other treatments. So it's hard, man. We're going to figure it out, and we're getting better, but it's not easy.
Yeah, absolutely. Thank you. Thank you, Lyle. Cindy, I just want to make sure before we move on to Phil, did we answer the questions that you had? And if
yes,
yes.
Okay, great.
Yes.
Right.
So Ed is asking me, can you hear me? Can everybody hear me okay?
Yeah.
Okay. Merit Sokovich recommended that Ed try to get to the USC Keck School of Medicine, and that it would take 3 months to build or grow his stem cells in a petri dish. And is that the typical type of path that this targeted medicine would take? And
yes, it takes a long time to get from the skin cells to the stem cells to then to the motor neurons. It may not take as long as 3 months, but definitely a couple of months, and then that's just the beginning, right? At that point, we really have to study the disease in the dish, and the information that we get out of there may take some time. But definitely, as a proof of principle, one could really try to knock down again the SOD1 gene with that mutation and see how the motor neurons behave. But typically the path, the timing is that. Now, I don't know if this is what NLORM is gonna do, because I don't think that that's the idea of NLORM, is not to study what happens in the Petri dish, is really to design these, as I call these antisense oligonucleotide, as I call this reverse gene therapy, that will target that specific gene and will knock it down. And I don't know timing about that.
Yeah.
So I put in the chat. The website for NLORM. I don't know more than I told you, but it's worth, I think, exploring it. just go online and see. Thank you.
Great. Thank you. All
Okay.
right, Philip, I don't know what took longer to get your boards or for you to wait in this to get your hand up. So the floor is yours. You have a great question for us.
Oh, thank you. It's not really so much of a question, but more like I would say a suggestion from my own experience. Someone mentioned that education was not enough, and I agree with that. However, more education in some instances can be helpful. When I went and got diagnosed, my doctor didn't tell me anything. I had to find out everything on my own. And I was diagnosed in June, so when October came, that's when I was finally placed into a clinical trial. But before I got to the trial, I spoke to maybe 2 or 3 different people and organizations just to get that access. So that 4-month time span could have possibly been shortened if my doctor had educated me on all the social issues, scientific updates, and different things happening for the cause. So I feel like the lack of education that I received prolonged some of the time that shouldn't have been in the first place if I was enlightened in the first place. So I feel like getting all the neurologists like on the same page in regards to patient education, as opposed to getting diagnosed and told you have X and X time left, and actually feeling like what you're being told is taken seriously. So if my doctor told me more, like I didn't know about the Healy trial. I did pre-med in college and I did nursing school, so I knew what ALS was from the moment the doctor told me. So there was no need for explanation. So I already thought my life was over. But after doing the research that I've done, it gave me hope. Meeting people at the clinical trial who also brought me to this meeting today, which I'm thankful for. It even gave me more hope. And, being told you have ALS, I would say 2 or 3 years ago was the equivalent of being told you have HIV back in the 80s. It's like your life is over. So if there was like a program that was implemented for all healthcare providers to request samples, such as CSF, skin samples for stem cells to help differentiate, like it was stated early in the conversation, and doctors really pushing the whole idea of getting treatment and educating the people like myself, because in actuality, it's all up to us at the end of the day. And that's one thing that bothers me. Before, if I had a problem, I go to the doctor tells me what's going on, doctor fixes it. It bothers me that it's not as simple as that. And there's a mixture between science and a sociological issue that is causing this whole thing to be cumbersome. So if more doctors, I feel, were on the same page as pushing education as opposed to writing patients off, which I felt was happening to me, I feel things would be better. Because like I said, I would not be sitting here today if I didn't take it upon myself to look things up, as opposed to my doctor not telling me anything. So everyone that created this program here today, and everyone that's done everything that you've done to get here today, I personally thank you for. And my doctor didn't know anything. He didn't. And
Well, Phil, I think you have just defined where you want your advocacy to be, which is in healthcare provider education, and person being now a nurse who has just passed his boards, those who joined us late, passed his boards today, so a round of applause. And yes, but you've defined where you want to put. So, you know, appreciate everyone who's come here and your questions and your challenges to us. Here in this milieu, you're preaching to the choir when you talk about education. I think this group of faculty, we are so passionate about educating other healthcare providers and making a difference to people who have to live with ALS and accelerating progress. We understand the urgency. While we understand and we're sympathetic, it is true we're not living it. And you guys are all, you know, you've lived it. And